It's my pleasure today to present a bit of a different tack to PTA, because I think we're going to be talking about PTA a lot, but also to give you a bit of context of some other Phytophthora issues that we've got in New Zealand forests. My name is Ari Williams, I'm based at Scion, and here we go. Okay, as Peter mentioned, many of the Phytophthora declines that we're observing, and we and colleagues internationally are observing worldwide, involve many Phytophthora species, and most are recognised by the main driver of that decline, PTA being the driver of cowering. But there are other Phytophoras in the system. Phytophthora cinnamomum, multivora cactorum in Antarctica. Oh, gee, I could go on. Okay, but in New Zealand we've got some interesting things. We've got a group of organisms that live in the soil. They spread, as Peter and others have said, by soil movement, in water, and through human spread. We then have these interesting ones which like hanging out at the top of the tree and live on the leaves. And interestingly, it was from here in New Zealand that Phytophthora captiosa and Phalanx were first identified on the leaves of eucalyptus trees by our colleague Margaret Dick. And so they're predominantly in eucalyptus plantations. But then more recently, we've noted Phytophthora pluvialis is also present on needles, and so again in the canopy, but on Pinus radiata, and I'll discuss that a bit today. And then we've got Phytophthora canovii, which likes to live everywhere really, and so it's on the roots and the leaves of trees. And if we look at its family tree, it looks like it's actually originated here in New Zealand, so that's kind of interesting. Most of the others we think have been introduced at one point or another. So today I'm going to focus a bit on these two down the bottom here, Phytophthora pluvialis and Phytophthora canovii. Now, so it's only in the last few years that a new needle disease was starting to be recognised on Pinus radiata plantations, predominantly on the east coast of the North Island. And following some reports internationally of other Phytophoras that were all of a sudden up in the canopy, the team at Scion in our diagnostics lab started to look to see whether or not Phytophoras might be involved. And when they looked into this disease, they noticed this overall red tinge to the trees in the lower canopy. Looking a bit closer, the needles on the lower side of the branches were starting to flag and go red and basically look sick. And when you look even closer, we see this characteristic banding, which did look quite similar to the Phytophthora that they'd seen in Chile. Fortunately, we didn't have that one. We isolated from that this new species, Phytophthora pluvialis. And so here I've got some pictures of, can you see the cursor? Yes, you can, good. Right here, these are the little sporangial sacs of Phytophthora pluvialis on a needle, Pinus radiata needle. Now, when we started to look into Phytophthora pluvialis a little bit more, we found some evidence that it's most probably been introduced relatively recently, and most probably from Western North America. Colleagues over there had isolated this species from their natural forest in mixed Douglas fir stands, and they found that they've got a wider diversity of Phytophthora pluvialis over there than what we have here, suggesting that it's actually been an introduction into New Zealand. And yeah, so that's quite interesting. Over there they don't observe that it causes disease, again suggesting that it might have evolved over there, whereas here it's causing new symptoms. Now, so at Scion, and obviously we do a lot of work with Pinus radiata and with the forestry industry who are a bit concerned about this new disease. And so we started to look at how much of an impact it might have across the forestry stands of New Zealand. So here's some analysis that we did on the development of lesions with Phytophthora pluvialis and canovii on different clones of radiata. And you'll see that there's some variation there across the different clones that I've got laid out from A to T. And so why have I got canovii there as well? Well, Pluvialis seems to be the main cause of red needle cast, but canovii is there in the system as well. And when we start to put our hats on as scientists of there being lots of Phytophoras out there in the system, what happens if a new Phytophthora comes in that's completely new? Are we going to see similar patterns between Phytophthora pluvialis to Phytophthora X that comes across the border? And so we do want to start asking these questions of, well, under the right conditions, would radiata be susceptible to any Phytophthora? And this analysis comparing Pluvialis and Canobii, when we actually, under lab conditions I must stress, when we compare the susceptibility to the two species, you can see that they're fairly well correlated. So that suggests that there's capacity within the Pinus radiata population to select for resistance to Phytophthora pluvialis and most probably other Phytophthora diseases. Now that's really provides us with quite a lot of hope, if you put your cowrie hat back on for a moment, that perhaps if we can select for resistance to Phytophthora taxon agathis, that we'll actually be protecting trees against the other Phytophoras that are out there, the Phytophthora cinnamomoes, the multivoras, that might not be having a lethal impact on cowrie, but might be sitting there and just decreasing the health of the trees. So I think that's quite hopeful. Just a few things about Pluvialis is that it results in the defoliation of trees, which is obviously quite contrasting to PTA affecting the roots. It affects the lower crown predominantly, but it's only found in the needles. So we don't see any typical lesions and the bleeding lesions up a stem like we see with PTA. And we don't have any evidence of the infection entering into the branch of the tree. And most significantly for the forestry industry, we don't have any evidence of there being contamination or colonisation of logs. So, it's aerial. It has a limited host range at this stage on radiata pine and Douglas fir. Its expression is often very patchy. Unlike cowrie, where you've got a standing dead stag that's slowly dying over time, once the needles of radiata have fallen to the ground, there's actually no disease to see per se. It's all fallen away. So that raises a few issues in terms of studying the disease. We believe it's a recent arrival, and based on that, it's then established very quickly across the North Island and northern South Island of New Zealand. Which does not bode well if there happens to be a new aerial Phytophthora introduced into New Zealand, because our evidence from this example would suggest that it will spread very quickly and get established quite quickly. New Zealand has a nice temperate climate with lots of rain, whereby aerial Phytophoras have the potential to get established really readily. So I think it's important for us to keep that in mind. And it responds to breeding and selection. Now, Peter presented a variation on this data, but it's essentially the same data set, looking at the number of new Phytophthora species that we are becoming aware of over time. And you'll see that not only do we have an increase in the overall number of Phytophoras, but we're seeing an increase in both the soil-borne and airborne Phytophthora species. And to us Phytophthora researchers around the world, this is of great concern. The large epidemics of sudden oak death was aerial in its distribution, and it spread widely throughout North America and Europe just in the last 15 years. Phytophthora pluvialis is a new one to add to that list. As I have mentioned, we then have the compounding impacts of multiple species. We need to know what stress the other Phytophoras are having in the system. Right, but there's good news. Despite the fact we have groups of Phytophoras that are soil-borne and groups that are airborne, and they're on different hosts and in different places, there are still some fundamentals of Phytophthora biology that are the same. And I think it's important that we focus on those fundamentals when managing across species. They are predominantly spread by human interactions. The spread across sites are then fundamentally the same. If you're a soil-borne species, you'll move by the movement of soil and humans. If you're an airborne species, then you're going to move in air movement, water splash and fog. There is lots of evidence across different Phytophthora species that plants respond well to breeding and selection. We've seen that with Phytophthora cinnamomi in Australia, Phytophthora lateralis in North America, and the evidence that I've shown you here today with Phytophthora pluvialis and Canovia. There seems to be a fairly uniform response to phosphite in many systems. Now, it's very important that we note that that needs to be tested and optimised, for an individual system before it's rolled out into natural environments, but it certainly provides some hope that in high-value sites we've at least got something that we may be able to do. The fundamentals of biosecurity principles remain the same, of keeping clean in the green and not to spread the red, which is what they say in Western Australia. When you map out the green sites that you saw on Peter's chart of Western Australia, we want to keep those green bits green and the red bits, well, they're going to be red anyway. But the good thing is that these fundamentals mean that we actually have a really sound basis for applying adaptive management principles. And what I mean from that is by taking the existing knowledge, and that existing knowledge is both from the scientific literature and international literature, but also from the observations that have been made and the knowledge of the forest here in New Zealand. We then need to apply best practice management. What can we learn from internationally about the management of these diseases? How does it then apply to the New Zealand situation? And what are the priorities that we need to get on and start doing? We then need strategic and targeted research that actually informs management. It's all well and good for us to sit in our laboratories and work out some of these fundamental things, but it needs to get back onto the ground and inform management. Otherwise, most of us would probably go to the beach instead. So we then need to have a constant process of review as to how well our management and science is actually working together, and whether or not we're actually interacting at the ground level, and that of course includes community. So, as was mentioned, we have some funding to actually look at some of these fundamentals, but we're not completely removed from the realities of management on the ground. Our overall project is targeted at exploiting, must be an important message, exploiting the fundamental similarities between Phytophthora species. We need to understand the genus as a whole, and then we need to target management, prescriptions and research across the Phytophthora diseases. Otherwise, we're going to be micromanaging individual diseases across the landscape. These target things like the broad host resistance, chemical control strategies, but also novel control targets that may have a broad application. So Scion is the lead agency on this new project, Prosperity from Trees Protecting from Current and Future Disease. We're looking at the response of key host plants, including Pinus radiata, kauri and apple, to Phytophthora diseases, to understand the fundamental similarities and difference between these pathogens. While Scion is taking the lead, we are also collaborating with many of the people in the audience here, including Landcare, Plant and Food, University of Auckland, Massey University, and international groups, including CPSM, who we've got here today. We then also have the cooperation from Auckland Council and other partners. So, on that note, I'll hand over to Nick.